And for our next talk, this is actually a student lightning talk. In the past couple of years, we've introduced these lightning talks throughout the program, and they've been really well received. So it gives us a chance for students to showcase their work. And so I'd like to invite Yin Peng to give the next talk here. And that should be good.
So hello everyone. So my name is Yunpeng Gao. So today I'm giving a lightning talk about my project. The topic is the geospatial tool to locate septic system and estimate the septic derived phosphorus load to. And since my colleague Evan has already given the background and contaminations of septic system, so I'll jump directly to the objective. So for my objective, so I need to first improve the geospatial tools for spatially locating the individual septic system across Ontario, Lake Erie, and Lake Simcoe Basin. And this tool was initially developed in 2020 and needs to improve because the update of data and improve the accuracy. And secondly, the tool is to estimate the phosphorus load to the tributaries based on the physical and the socio-economic factors. And these are the factors which Evan is considering. And this project is funded by Ontario Ministry of Environment, Conservation and Parks. So for my methodology, so I first need to find the rural houses with septic system. And to find those houses, I first use LiDAR data to do a classification to filter all the objects above the ground. So you see here is the here's the raw LiDAR point cloud data. You can see all the stuff above ground are the buildings and trees. So first step is we retrieve those features, and then we calculate another value called NEVI from the multiple aerial image. And this value can help us to distinguish the buildings and trees since it's very sensitive to the green color, and the green color comes from the chlorophyll A from the leaves. And after that, we use the machine learning method to classify all the stuff, and finally we got 5 class. And you see here the dark red ones are the buildings. And then we place the septic system near those buildings we identified. For my result, I got, so here's the 2 density map of septic system. So for Lake Erie Basin, we got more than 122,000 septic system. And for Lake Simcoe Basin, we got more than 28,000 of septic system. And in addition, I developed online tools to first identify the number of septic systems within the region, and then to estimate the septic derived P-load from the region. And it also can show the physical and the social economic characteristic from this, for the septic system in the region. And I just made some screenshot of the tools. So first here is the map of all the septic system, and here is the total number of the septic system on the map. And then we have a worst case phosphorus load calculated. The worst case is we assume no attenuation were happening for the phosphorus load, but in actually there is attenuation. So that's why we need advanced statistical model to have a better refined phosphorus estimate. And also we can show the environmental factors. For example, here we are showing the permeability, but also we can change to the tile drainage area. And also we have the socio-economic factors, for example, the median household income, the household age, and the home construction period. And also I add functions to kind of a small filter here. It can filter to the tertiary and the quaternary watershed. For example, today we're in Waterloo, and we are inside Upper Grand River. So if we turn on the filter and select the Upper Grand River, we can the map will automatically zoom in and pan to the Upper Grand River. And notably, so those values are all dynamic, and it's changing while we are kind of moving, zooming in and out or pan to any regions. And also we have the filter for the quaternary watershed. For example, we are in Waterloo, and our closest watershed is Conestoga River watershed. And here is the septic system inside the Conestoga watershed. And to summary, so new geospatial tool was based on the remote sensing method. It's developed to identify the individual septic system locations. And the online tool is developed for the users to determine septic system numbers and the phosphorus load in a given region. And my next step is to apply the statistical model from previous presentations to calculate the refined septic derived phosphorus load in the online tool. And that's all my presentation. Thank you very much.
Okay, thank you. You can stay up here, Evan, if you want to come back up. We do have some time for questions. If anybody has any questions for Evan or Yim-Peng. Takers. Oh, we have a question at the back there. Steve's going to come with a mic.
Steve, can you speak on whether you saw any correlation between either the DNA marker or the other one you used with any of the nitrogen species in your samples?
I specifically did not look at the nitrogen data for correlating with the markers, but if we're looking at in terms of the relationship to nutrients, whether that be phosphorus, or specifically phosphorus, in our case in the Great Lakes, the HF1A3 has a decay, which can mimic the relation or the behavior with phosphorus in the environment a lot more than the acesulfame, which is highly conservative. So it's not, the acesulfame doesn't really account for that adsorption or attenuation that can occur with phosphorus or associated nutrients in the plume. I don't know if that answers, but.
Hi there. Quick question with the artificial sweeteners. Are there any considerations from sample collection to when you actually have those analyzed? I.e., if the samples were frozen, would that still be? Would you still get accurate results if you were to analyze those frozen samples for artificial sweeteners?
Yeah. So we collect in the field, and we freeze upon returning to the lab, and then they're thawed and then analyzed accurately. Yeah.
All right, one more quick question.
Yeah.
Because of the fact that I think it was nanograms per liter was the accuracy for the natural sweeteners, do you need a pretty large volume of sample to be able to run the analysis?
I'm looking at the person who runs my samples, and she said, no, a small volume. Yeah.